Okay, here we go. I'm really sorry, this is a really long question and it's on the kidney, but you can always fast forward it for the bits that you got right. So, here we go. So this is component 3, Mock 2, question 5, the kidney. So, glomerulus and Bowman's capsule of the kidney nephron are the site of ultrafiltration. Image 1 shows the main structure, so we've got the afferent, and it's really obvious on this, that's wide, and the efferent narrow. We've got the glomerulus and Bowman's capsule, so that's a diagram of the corpuscle. And we've got image 2, got to work out what this is. That looks like a nucleus to me. Could be, it looks like a stray mitochondria in it. So this is obviously a cell of some description. There's a bit of a gap, and then these little things here. So they are, I'm thinking, they must be podocytes. Cells with feet, how lovely. We've got the endothelium, so that must be lining the blood vessels, so that will be a fenestration. And then you've got the basement membrane in between. So now I know what's going on. So we've got blood, basement membrane, podocytes around the outside, and we've got a scale bar, and we've got a space for a calculation, so we're going to have to do something with that. On image 1, label with a P the part of the nephron where the structure shown in image 2 will be found. So where are the podocytes? They're kind of there on the outside of the... So I'm going to put just a line there. I'm going to use a ruler. Why wouldn't you do that? And label it P like I've been told to do, following the instructions. Using the scale bar on image 2 only, calculate the magnification of this electron micrograph. So I'm going to do magnification equals I over A. My actual size is 500 nanometers. Crikey. So measuring my scale bar, that's 14 millimeters. So I've got 14 millimeters divided by 500 nanometers. So I like to deal with big numbers, so I'm going to convert my 14 into nanometers. So 14, 3 zeros to micrometers, 3 zeros to nanometers. And then I'm going to divide by 500. I'm going to have to do a little bit of a cancelly thing here because my calculator's so useless. There we are. Cancel those zeros out and hope it can cope. 14. Now I've got 4 zeros. 1, 2, 3, 4. Is that right? 1, 2, 3, 4. And 4 divided by now 5. 28,000. And again, usually I would check my calculations again and again and again because I am not very good at it. So, the effective pressure which forms the glomerular filtrate is due to several different pressures. Hydrostatic pressure in the glomerular capillaries, so that's an outward force. Osmotic pressure due to the plasma proteins, that's an inward force. And pressure of the fluid in the capsule, so that would be an in. Oh, it tells you that. Pressure forming glomerular filtrate is the hydrostatic pressure minus osmotic pressure minus interrenal. In a healthy adult, the osmotic pressure of the plasma is 4, the interrenal pressure is 2.6, and the hydrostatic pressure is 8. So, calculate the pressure. Pressure equals 8 minus 4 minus 2.6, which equals 1.4. I can do that in my head. And then let's remember the units, kilopascals. Use the information to suggest how a low protein diet could affect the rate at which glomerular filtrate is formed. So, osmotic pressure, if we've got less protein, then the osmotic pressure will go down. Or, if you prefer, water potential will go up. And therefore, that number will be less. So So say we dropped it to 2, that would mean that it was 8 minus 4.6, which would be 3.4. So the pressure would increase, okay? So I'm just going to write that down. So somehow I've got to explain how that works. So what do I know? Less protein in the plasma would lower the osmotic pressure, water potential would be higher, and therefore the pressure forming the filtrate would increase and what are we being asked about? So rate at which filtrate is formed and increase the rate of filtrate formation. I could have put the word glomerular in there and that would have been probably nicer and more accurate. Anyway, not to worry. Okay. The pressure forming the glomerular filtrate can be regulated. The walls and the muscles in the walls of the afferent and efferent arterioles are under hormonal and nervous control. Describe how changes in the afferent and efferent arterioles could increase the pressure. So, just go back to my diagram. We've got a wide afferent and a narrow efferent which raises the pressure. So if we made that one narrower, that would increase the pressure. We put more of them on the hose pipe. Or we could switch the tap on more and make that wider and deliver more blood in with the same narrowness. So, okay. So, dilating the afferent arteriole would increase blood into the glomerulus and contracting the muscles in the efferent would restrict. Blood leaving and increase pressure. In diseases, moving on to different thing now. In diseases such as diabetes mellitus and glomerulonephritis, membranes in the glomerulus and Bowman's are damaged and they become more permeable to proteins. Suggest 2 reasons why the proteins are not usually reabsorbed back into the bloodstream. So now we're talking about selective reabsorption, and we're obviously moving down into the proximal convoluted tubule. So why you wouldn't get them reabsorbed, obviously they're very big, so they won't diffuse, and you can't have carriers for them. So proteins are not reabsorbed. I'm going to say where it is. Why wouldn't you? Might be worth a mark. As they are too big to diffuse and they're not supposed to be there. Oh no, so there won't be any specific carriers for them because they're not usually there. That was nice and easy. Okay. Where are we now? Oh, the urea concentration increases as the filtrate moves along the proximal convoluted tubule, but the mass remains constant. Explain why the urea concentration increases. I don't know how many times we've answered this question. So when they're asking about concentrations, you also need to be talking about water. So the concentration increases as water is reabsorbed in the proximal convoluted tubule. It just comes up time and time again. Okay. If the concentration of urea along the proximal convoluted tubule is plotted on a graph, the line of best fit would follow the equation shown below. So y, which is this, equals m, which is the gradient, times x, which is the value along there, plus c, which is the intercept of the y-axis. So, concentration of urea at the filtrate at the start is 20, so that's your C value. So, you know, at the start is 0, so your X value is 0, so no matter what so it's just Y equals C. And then the gradient of the line needs to be 0.2. Use the values provided to calculate the concentration of urea at the end of the tubule and draw a line on the axes below for the concentration. Okay, so at the end of the tubule, X equals 100. Have they left us a gap to do it in? Right, and we were trying to work out Y so that we know, our X value. So Y equals M, gradient, 0.2, times X, 100, plus where it intercepts the y-axis, which is at 20. And here we go. 0.2 times 100 is 20. 20 plus 20. So my plotted point is going to be at 140. Need to do that carefully, blimey. I think it's because my eyes are old. That's the line there. So that's my point. And can't take it beyond that point because I've no data. The end of the tubule, anyway, it can't go beyond the end of the tubule, can it? A nice straight line between those two things. So just looking at the mark scheme, it says point drawn at 40 and 100, line drawn with a ruler between the 2 points. This is why you need a ruler in your exam, one of the reasons. You'll be measuring things, you'll be drawing lines. Okay, so here we go, still in the proximal convoluted tubule. I can tell because I can see a brush border. It shows a section through the wall of the proximal convoluted tubule. So we've got Z, they're the microvilli, and X, that's the nucleus. I'm guessing these will be mitochondria, they're sort of mitochondria shape. Explain, so give a reason why there are so many of the organelles labelled X in the cells shown. So they've not told you what it is, so I'm thinking that there might be a mark for mitochondria. So X are mitochondria, which release energy in the form of ATP, and I don't know what my mark's going to be, whether it's going to be energy or ATP, for, why do they need them, active transport. Should have planned that before I started. We'll put that down there, because I don't know what they're going to give these marks for. From aerobic respiration, always worth a punt. Sometimes you won't get anything for putting just respiration. So mitochondria just do aerobic. Explain the presence of structures Z. So again, I'm going to say what they are. Z are microvilli, which increase surface area, and again I'm just going to go for absorption. So say what it's for, that's an explanation. And switching again, lungfish. Survive the drying out of their habitats by burrowing into the mud and decreasing their metabolic rate. Explain why, when in water, they release the products as ammonia, so water ammonia, and in dried out mud, they use urea. So again, I'm just going to use this bit at the bottom to do a little bit of a plan. So what do I know about ammonia? I know it's toxic, and I know it needs large volumes of water to eliminate it. That's quite unusual, isn't it? Fish making urea. Anyway, and then I've got urea, less toxic. I'm just going to write storage after that, because I think that might be important. If you're in mud, you want to produce something that you can actually hang on to for a bit until you've got some water to get rid of it. It's less soluble, needs less water, so you can conserve water. And again, if you're in dried out mud, I think that's what you're going to need to do. Okay. And I think we need to compare them, because explain why they do one but then they do the other. That's a sort of comparison thing. So we'll start with the properties. So ammonia is more toxic and more soluble than urea. As it is toxic, it needs to be removed, and as it is soluble, this requires more water. Let's turn to why they do it. So in dried out mud, there is less water. I don't think you can say there's no water, there's bound to be a little bit there. So the lungfish needs to conserve water. Urea needs less water to be excreted. And there's less can be stored without harming the fish. So remember, we've got that sort of idea of variability of excretion. Oh my goodness, I think Dr. Saville will have to split that into two.